Assalamu alaikum dear students. I am Dr. Shahjabeen and today's lecture topic is classification of nerve fibers. These are the learning objectives of this lecture. At the end of the lecture you would be able to define neuron and describe its structure, discuss methods of classifying nerve fiber, explain nerve fiber classification on the basis of diameter and conduction velocity, along with the sensory nerve classification that is also known as numerical classification, and list the properties of nerve fibers, describe causes of injury with the degrees of injury to nerves, explain three types of degenerative changes in neuron including Wallerian, retrograde and transneural degeneration. You would also be able to describe regeneration of nerve fiber including its criteria and the stages of regeneration. Neuron or nerve cell is the structural and functional unit of nervous system. These neurons are linked together in highly intricate manner so that our body is aware of the changes in the environment as well as in our own body and can produce appropriate response to the changes. A neuron primarily consists of a cell body that is also known as soma and the neurites or the processes, and these neurites or processes are of two kinds, dendrite and the axon. These neurites or the processes are also known as nerve fibers. Here in the picture you can see a cell body which is soma, also known as perikaryon. This is irregular in shape, having a centrally located nucleus, and this nucleus of a neuron is devoid of centrosome. So the nerve cell cannot multiply like other cells. The cytoplasm of this neuron is known as neuroplasm, and it contains nissl bodies or nissl granules. You can see these smaller granules, and these nissl granules or bodies are present in the soma and the dendrites, and these are absent in axon hillock and the axon. This is the axon. These nissl granules or bodies contain ribosome that are attached to the rough endoplasmic reticulum. Hence, these are concerned with, yes, protein synthesis that is needed for the maintenance and repair and for the production of neurotransmitters and enzymes. One important point to be noted here is during injury of neuron, these nissl bodies or nissl granules fragment and disappear by a process known as chromatolysis and reappear after regeneration. The neuroplasm also contain neurofilaments, you can see, and the mitochondria and Golgi apparatus that is not shown in the diagram. And the second thing, the neurites one, are the dendrites. This you can see the processes, the shorter processes are known as dendrite. And these dendrites are known as receptive processes as they receive impulses from the neurons or the sensory neurons or receptors, and transmit these impulses towards the nerve cell. While the larger, usually the longer process is known as axon. This is axon. And this axon is the process that transmits impulses away from the nerve cell. The axon terminates by dividing into a number of branches, and these branches end in a knob shaped structures known as synaptic knob or the terminal buttons. These terminal buttons have micro vesicles in which chemical neurotransmitters are stored. Here you can observe one more thing, that this axon is covered by a sheath, and this sheath is known as myelin sheath. What is a myelin sheath? It is a thick lipoprotein, that is the combination complex of protein and lipid, that is present around the axon. Here you can see in the picture, this is the axon of the neuron, and it is covered by a sheath that is known as myelin sheath. And these axons are known as, that are covered by the myelin sheath, are known as myelinated nerve fibers. This myelin sheath is not continuous covering, and it is absent at regular intervals. You can see from the picture. And the area where this myelin sheath is absent is called as node of Ranvier. And the segment of the nerve fiber between two nodes, for example this and this one, is known as internode. This is known as internode. The process of formation of myelin sheath is known as myelinogenesis. And this myelin sheath is formed by concentric layers, deposition of concentric layers of proteins alternating with lipids. And the lipids may be cholesterol, lecithin, and cerebroside. The myelin forming cells are glial cells or the supporting cells. In the peripheral nervous system, Schwann cells form the myelin sheath, while in the central nervous system, that is in brain and spinal cord, oligodendrocytes form the myelin sheath. Here you can see in the picture, the Schwann cell is forming the myelin sheath, that is, it is the peripheral nerve. And here you can see the concentric layers of protein and lipids. It is actually formed by the wrapping up of Schwann cells around the axon. And in this picture, beside this, as you can see oligodendrocytes that are forming myelin sheath around the axons. And this would be, yes, this occurs in the CNS. In the peripheral nerve, the myelinogenesis starts at fourth month of intrauterine life, and it is completed in the second year after birth. The myelin sheath provides faster conduction, less energy consumption, and high insulating capacity to the nerve fibers. In myelinated nerve fibers, the impulses jump from one node to the another node of RANVA instead of traveling through an entire nerve. And this type of transmission of impulses is known as saltatory conduction. Here you can see in the picture. This is saltatory jumping manner transmission of impulses. So, in the myelinated nerve fibers, the impulses are conducted 50 times faster than in a non-myelinated fiber. And in this way, this myelin sheath also consume less energy. The myelin sheath restrict the nerve impulse within the single nerve fiber and prevents the stimulation of neighboring nerve fibers, thus providing insulating capacity to that nerve fiber as well. Now, if we see the internal detailed structure of axon, it has a cytoplasm known as exoplasm, and the axon is covered by a tubular sheath-like membrane called exolemma. This exoplasm is covered by exolemma, which is the continuation of the cell membrane of nerve cell or nerve soma. Exoplasm along with the exolemma is called as axis cylinder of the nerve fiber. Here you can see this is exoplasm and exolemma, and in the myelinated one, this is exoplasm and this is exolemma. Both of these are known as axis cylinder of the nerve fiber. Now this axis cylinder of the nerve fiber is covered again by a membrane that is known as neurilemma. This is the black color, that is neurilemma. This covering is known as neurilemmal sheath or the sheath of Schwann, as it is made up of Schwann cells. In non-myelinated fibers, look at the B. The neurilemma that surrounds exolemma. This is the neurilemma and it surrounds exolemma continuously. While in the myelinated nerve fibers, look at A, it covers the myelin sheath. Here you can see this neurilemma, black colored, is covering the myelin sheath. And at the node of RANVA, you can see at the node of RANVA, where the myelin sheath is absent, the neurolemma sheath invaginates and runs up to the exolemma. Here you can see. Now, what is the significance of this neurolemma sheath? In non-myelinated nerve fibers, the neurolemma serves as a covering membrane, while in the myelinated nerve fiber, it is necessary for the formation of myelin sheath, that is myelinogenesis. As in the periphery, the myelinogenesis is done by Schwann cells. Neurolemma is absent in central nervous system. So which glial cells forms myelination or the myelin sheath in CNS? Yes, oligodendrocytes. This slide shows how axons or nerve fiber organize to form a nerve. What is a nerve? Nerve is a compact bundle of axons or nerve fiber that are located outside the CNS. While in the CNS, the collection of axon is known as tracts. Here you can see in this picture, that is the example of a spinal nerve, that axons are grouped together to form a fasciculus. Here you can see this is fasciculus, and many fasciculi, here you can see many fasciculi arranged to form a nerve. Each axon or the nerve fiber is covered by a delicate connective tissue layer that is known as endoneurium, while each fasciculus, that is the collection of axon, is covered by a perineurium that act as a barrier to the passage of molecules, toxins into the endoneurium. The whole nerve is covered by a connective tissue layer that is known as epineurium. Epineurium limits the extent to which the nerve can be stretched by body movements or external pressure. Thereby, it protects the fragile axons inside the nerve. Now come to the main topic, that is classification of nerve fibers. Nerve fibers are classified by different methods. The basis of classification are their structure, their distribution, origin, their different function, and the secretion of different type of neurotransmitters. The most important one is the classification that depends upon diameter and conduction velocity of nerve fibers. We have two classification on this criteria. Number one is Erlanger and Gasser classification. It is also known as lettered classification. And the other one is Lloyd and Hunt classification, also known as numerical classification or sensory classification. Depending upon the structure, the nerve fibers are divided or classified into myelinated nerve fibers and non-myelinated nerve fibers. As the name says, myelinated nerve fibers are covered by myelin sheath and have greater conduction velocity than the non-myelinated ones, which are not covered by the myelin sheath. For example, slow pain fibers. Multiple sclerosis is an autoimmune disease in which the antibodies attack myelin, causing inflammation and injury of this sheath. Here you can see in the picture below is the damaged myelin, or you can say the damaged nerve of the multiple sclerosis as compared to the normal nerve above. So the loss of myelin sheath leads to failure of action potential conduction, and the person or the patient feels weakness, fatigue with the diminished motor and sensory functions. Depending upon the distribution of nerve fibers in the body, these are classified into somatic nerve fibers and autonomic nerve fibers. Somatic nerve fibers supply the skeletal muscles of the body and they are voluntary in nature, while the visceral or autonomic nerve fibers supply the viscera or internal organs of the body like heart and GIT, and they are involuntary in nature. Depending upon their Origin. These nerve fibers are classified as cranial nerve fibers and spinal nerve fibers. As the name says, cranial nerve fibers arise from brain while the spinal nerve fibers arise from spinal cord. Depending upon their function of carrying impulses, these nerve fibers are classified as sensory nerve fibers and motor nerve fibers. Sensory nerve fibers carry sensory impulses from different parts of the body to the central nervous system, and these are also known as afferent nerve fibers, while the motor nerve fibers carry motor impulses from the central nervous system to the different parts of the body. That is, they convey the motor effect from the central nervous system to the different parts of the body, and that is why these are known as efferent nerve fibers. On the basis of secretion of neurotransmitters, these nerve fibers are classified as adrenergic nerve fibers that secrete noradrenaline and the cholinergic nerve fibers that secrete acetylcholine. On the basis of nerve fibers' diameter and conduction velocity, Erlanger and Gasser classified the nerve fibers into three types: A, B, and C fibers. That is why it is also known as letter classification, as denoted by letters. This classification includes all type of fibers, that is motor, sensory and autonomic fibers. Here in the table you can observe type A fibers are the fastest fibers as their diameter, as their conduction velocity is ranging from, you can see, from 12 meter per second to 120 meter per second, and their diameter is more, that is from 2 micron meter to 20 micron meter. Remember, the velocity of conduction in the fibers is more when the diameter is more, while the duration of the action potential, or you can say the spike of action potential, and the refractory period, especially the absolute refractory period, is less when the diameter is more. So here you can see increase in the diameter, there is increase in the conduction velocity, while decrease in the duration and the absolute refractory period. Now, if you see type A fibers in detail, it is further divided into alpha, beta, gamma and delta fibers. Alpha fibers are of two types. They can be somatic motor fibers. That is, they supply skeletal muscles. Somatic motor fibers, they supply skeletal muscles. Or they can be somatic sensory fibers which sense proprioception. They are for proprioception, that is for the detection of position and movement of the body. Beta fibers carry touch, light touch and pressure. They are pure sensory fibers. Beta fibers are pure sensory fibers. Remember, A alpha fibers are both motor and sensory fibers. After beta fibers, we have A gamma fibers. They are motor fibers and they are motor to the muscle spindles. Muscle spindle are the stretch receptors present in the muscle. Now come to the last one, that is the A delta fibers. They carry fast pain and the cold temperature and the touch. Remember, only the cold temperature. Let's come to the type B fibers in this Erlanger-Gasser system. B fibers are preganglionic autonomic fibers, that is efferent and afferent fibers from the skin and viscera. Next is type C fibers. Type C fibers carry slow pain, some of the mechanoreceptors including crude touch and pressure and the temperature. In these C fibers carry both the temperatures, both cold and warm temperature. In their C or the sensory component, another type of C fibers carry the autonomic component, and these are postganglionic sympathetic fibers. Note one thing in this whole table, that all the A, B fibers are A, all including all the alpha, beta, gamma and delta, and the B fibers all are myelinated fibers, but the type C fibers are unmyelinated fibers, that is without myelin sheath. Another thing, as you move down in this classification from A to B, from A to B to C, the diameter of the fibers reduces. Here you can see the diameter of the fibers reduces. That is, fibers get progressively thinner, as a result of which their conduction velocities become lower or lesser. Here you can see, so you can conclude that the least conduction velocity is of C fibers and the maximum conduction velocity is of A fibers, and the A alpha, the fastest fibers, are the fastest fibers with the maximum diameter. Lloyd and Hunt classified sensory nerve fibers by a numerical system. Note, only sensory nerve fibers are included in this classification. Here you can see A alpha fibers. Only the sensory component of A alpha fibers are denoted as type 1 fibers. These are Roman numericals, and this is Lloyd classification. Type 1 fibers are again divided into 1A and 1B. 1A fibers innervate muscle spindle, and you know what is muscle spindle? This is stretch receptors present in the muscle, and they detect changes in length of the muscle. 1B fibers innervate Golgi tendon organs and they detect changes in the tension of the muscle. Both are A-alpha sensory component fibers. Now, A-beta fibers are known as type 2 fibers here in this numerical classification system, as A-beta fibers are all sensory and they carry sensation from touch, pressure, and they carry muscle spindle and flower spray ending sensation. Now, A-delta fibers are type 3 fibers that carry pain, fast pain and cold temperature along with some touch receptors. Now, where is A gamma fibers? A gamma fibers are not included in this classification, as A gamma fibers are motor fibers. Similarly, B-type fibers are not classified in this Lloyd classifications, as they are autonomic fibers. Now, C fibers are the type 4 fibers in this system. You know, they carry slow pain, both the temperature, cold and warmth, and crude touch. Now this slide shows the properties of nerve fibers. Most of these you have done in the previous lectures of action potential. Number 1 is excitability. It is the property of nerve fibers by which they respond and generate action potential. The stimulus may be mechanical, thermal, chemical, or electrical. Another property is conductivity, which is the ability of nerve fibers to transmit the action potential or impulse from area of stimulation to another area through nerve fiber. The conduction velocity is more in myelinated fibers due to saltatory conduction. Refractory period is the period in which the nerve does not give any response to a stimulus. It includes absolute and relative refractory period. During absolute refractory period, nerve does not show any response at all, whatever may be the strength of stimulus. It corresponds to the period from the time of firing level until 1/3 of repolarization. The ionic basis you have done in the action potential lecture. Next property is summation. That is, if two or more subliminal or subthreshold stimuli are applied within a short interval of 0.5 milliseconds, stimuli are summed up together and produce the response. This phenomena is known as summation. Another property of nerve fibers is accommodation. When a nerve fiber is continuously stimulated, the excitability is greater in the beginning, then later this response is decreased. This is because of continuous excitation causes continuous depolarization that inactivates sodium pump and increases potassium efflux. Now, another property of nerve fiber is infatigability. Nerve fiber cannot be fatigued, even if it is stimulated continuously for a long time. The reason is that nerve fiber can conduct only one action potential at a time and at that time it is completely refractory and does not conduct another action potential. The last property of nerve fiber is all or none law. This you have done in the properties of action potential as well. All or none law states that when a nerve is stimulated by a stimulus, it gives maximum response or does not give response at all. Now the topic of degeneration and regeneration of nerve fibers. Common causes of degeneration of nerve fibers are obstruction of blood flow resulting in ischemia, local injection of toxic substances, crushing of the nerve fiber, or transection of nerve fiber, that is a through and through cut. Sunderland classified the degrees of nerve injury into 5 categories depending upon the order of severity of injury. First degree, also known as neuropraxia, it is the most common type of nerve injury in which prolonged pressure over nerve results in the occlusion of blood flow, and because of the occlusion of blood flow, oxygen supply is reduced, that results in demyelination of the nerve fiber. In first degree injury, there is temporary loss of the function and there is temporary blockage of conduction of impulse. Here you can see there is, in the first degree, there is only mild demyelination has occurred. In the second degree of injury, that is also known as axonomesis, there is prolonged severe pressure and for a long period of time over the nerve, and there is severe damage to the nerve. But the endoneurium is intact. Here you can see the endoneurium is intact. What is endoneurium? Yes, it is the connective tissue sheath that surrounds each axon. As the endoneurium is intact, repair and restoration is complete. In third degree injury, here the damage is again severe so that the endoneurium is also interrupted. But epineurium and the perineurium are intact. Here you can see the perineurium. This is the perineurium, the connective tissue sheath over the whole nerve, and the epineurium, the connective tissue sheath over the fascicle, is intact while the endoneurium is interrupted. Here the recovery is poor or incomplete. In the fourth degree of injury, the damage is more severe. Even the epineurium and the perineurium are also interrupted. You can see from here. Fasciculi of the nerve fibers are disturbed and disorganized, and the regeneration is poor or incomplete. While in the fifth degree of injury, there is complete transection. You can see from the picture of the nerve trunk with loss of continuity. Useful regeneration is not possible unless the cut ends are rearranged and approximated quickly by the surgery. Third, fourth and fifth degrees of injury are known as neurotomesis. Degenerative changes are the pathological changes due to any injury. When a peripheral nerve is injured, the degenerative changes occur in the nerve cell body, nerve fiber of same neuron, and the adjoining neuron. And the degenerative changes are classified into Wallerian degeneration, retrograde degeneration, and transneural degeneration. Wallerian degeneration are the pathological changes that occurs in the nerve fiber distal to the nerve injury. Wallerian degeneration starts within 24 hours of injury. First, the axis cylinder swells and breaks up into small pieces. What is axis cylinder? It is the exoplasm of the axon along with the exolemma. After few days, the broken pieces appear as debris in the space occupied by the axis cylinder. Here you can see the debris. Myelin sheath is slowly disintegrated into fat droplets, and this occurs from 8th to 35th day. Here you can see the fat droplets, the yellow colored, these fat droplets of the myelin sheath. Neurolemmal sheath is unaffected, and you know the neurolemmal sheath is formed by the Schwann cells. It is also known as the Schwann sheath. So the Schwann cells multiply rapidly. Here you can see the Schwann cells are proliferating. Now the macrophages invade from outside and remove the debris that is formed by the axis cylinder and the fat droplets, disintegrated fat droplets of the myelin sheath. When the debris is removed by the macrophages, the neurolemmal tube becomes empty and later it is filled by the cytoplasm of the Schwann cell. All these changes takes place for about 2 months from the day of injury. Retrograde degeneration are the pathological changes which occur in the nerve cell body and axon proximal to the injury. Changes in the nerve cell body start within 48 hours. First, the nissl granules or the nissl bodies disintegrate into fragments by a process known as chromatolysis. Here you can see in the picture, there is the disappearance or disintegration of the nissl substance. Golgi apparatus is disintegrated also. Nerve cell body swells due to accumulation of fluid and becomes round. Here you can see the swelling of the soma. Neurofibrils disappear, followed by the displacement of nucleus towards the periphery. Sometimes the nucleus is extruded out of the cells. Now the degenerative changes that occur proximal to the injury. Here you can see the picture. This is the site of injury. In the retrograde degeneration, the degeneration or the pathological changes occur proximal to the injury, and these changes are similar to those changes occurring in the distal end, and those distal changes or degeneration are known as Wallerian degeneration. You have done it. It is sometimes observed that changes resulting from axonal injury are not confined to the injured neuron but extend to other neurons with which the injured neuron synapses. This phenomena or the type of degeneration is known as transneuronal degeneration, and the degeneration can extend through several synapses. Here you can see in the picture there is the site of damage, and distal to it WALLERIAN degeneration you can see, and proximal to it that is the retrograde degeneration. And you can observe here that the adjacent neurons showing degeneration, and this is known as transneural degeneration. Here is the retrograde one, and you can see at the end that is the anterograde transneuronal degeneration in the adjacent neuron. For example, this degeneration occurs in the cells of the lateral geniculate body due to the sectioning of the optic nerve. The term regeneration refers to the regrowth of lost or destroyed part of a tissue. The injured and degenerated nerve fibers can regenerate. The regeneration starts as early as fourth day after the injury, and it is completed in about 80 days. For the regeneration, gap between the cut ends of the nerve should not exceed 3 mm. Neurolemma should be present, and as neurolemma is absent in CNS, the regeneration of nerve does not occur in CNS. Nucleus must be intact, and if it is extruded from the nerve cell body, the nerve is atrophied and the regeneration does not occur. Two cut ends should remain in the same line, and regeneration does not occur if any one end is moved away. Now, how the regeneration occurs in the nerve fiber. First, some pseudopodia-like extensions grow from the proximal cut end of the nerve. These extensions are called fibrils or regenerative axonal sprouts. Here you can see in the picture, after 2 weeks of injury, you can observe WALLERIAN degeneration and chromatolysis. Afterwards, below it, you can see axonal sprout. Fibrils or the sprouts move towards the distal cut end of the nerve fibers, here from the proximal towards the distal. Some of the fibrils enter the neurolemmal tube of distal end and form the axis cylinder, that is the exoplasm and the axolemma. Now the Schwann cells line up in the neurolemmal tube. You know the neurolemmal tube that was formed when the debris was phagocyted by the macrophages. Now the neurolemmal tube actually guide the fibrils into the tube, and these Schwann cells also synthesize nerve growth factors which attract the fibrils from the proximal segment. Axis cylinder is now fully established after 3 months of injury inside the neurolemmal tube. You can see here that after the 3 months of injury, there is successful nerve regeneration, and the myelin sheath afterward is formed by Schwann cells slowly. Myelination is completed in 1 year, and the diameter of the nerve fiber gradually increases. However, the degenerated nerve fibers obtains only 80% of the original diameter, And here you can also see that in the nerve cell body, the Nissl granules appear as well as the Golgi apparatus. And the cell loses the excess fluid and the nucleus occupies the central portion. Though the anatomical regeneration occurs in the nerve, functional regeneration or the recovery occurs after a long period.
Thank you, students. This is my email address for your queries and the reference books names.